Just going to wait a few more minutes here for everyone to file on in here. Okay, it looks like things have stabilized. So I just want to welcome everyone here. Thanks so much for attending today's Watson Armour seminar. My name is Matt Nelsen, and I'm a research scientist here at the Field Museum. The Field Museum acknowledges that it resides within the traditional homelands of the Three Fires Confederacy, the Ojibwe, Odawa, and Potawatomi. And I recognize and I'm grateful for the original peoples who laid the foundation for the city of Chicago and for the diverse indigenous nations that reside in Chicago today. So today I am thrilled to introduce Dr. Daniel Park. Daniel earned his Ph.D. from the University of California, Davis, where his dissertation work focused on trying to understand biological invasions from an evolutionary perspective. He then completed 2 postdocs and worked as a research associate at Harvard and the University of Arizona, and is now an assistant professor in the Department of Biological Sciences at Purdue University. His work has appeared in numerous prestigious journals, including Nature, Science Advances, PNAS, Ecography, and New Phytologist, I feel like his work just really utilizes and develops some extremely exciting and innovative approaches that utilize citizen science, collection data, and artificial intelligence to better understand how natural communities are impacted by global change. I want to thank Dan for joining us today, and I'm really looking forward to your talk. Dan?
Thank you for the wonderful introduction, Matt. And thank you for the invitation. I am I'm honored to be here and have a chance to talk about my research and share some of my results. All right, I'm going to share my screen here. Can everyone see my screen?
Looks good.
All right, great. Okay, again, thank you everyone for coming. Today I'm going to talk about plants in time and the patterns and consequences of intra and interspecific phenological variation. So phenology refers to the timing of life history events. What is that exactly? It is when events such as flowering or fruiting or leaf out in plants happens. In animals, it could be the timing of migration or emergence. And the timing of these events are very important to an organism's survival and fitness. For instance, if a plant emerged and flowered in the wrong conditions, there would be a very limited chance of successful pollination, not to mention survival. However, plant phenology has been rapidly changing of late. For instance, this photo was taken from an article in the Wall Street Journal a year or 2 back. And this article reported that cherry blossoms are in bloom in Central Park in New York in January. In general, cherry blossoms are symbolic of spring, not winter. And so this is an oddity, but you've probably heard of similar examples where plants seem to emerge and flower at odd times in the year or are persisting longer than usual. Okay, something's wrong here. Okay, so in accordance with global warming, many plant species, both wild and cultivated, have been displaying advanced phenology. This means that these events such as leaf unfolding or fruiting or flowering or shooting are happening earlier than before. Here in this graph, you can see that these events are advancing. The dots are all under 0, indicating negative responses to rising temperatures. This meta-analyses by Menzel et al. Looked at 125,000 observational series in Europe and basically found that the majority of systems and phenological events are, again, negatively correlated with temperature, meaning warmer climates lead to earlier events. The accurate timing of life history events is critical to an organism's survival and fitness, but it's also because plants they form the basis of most, if not all, terrestrial ecosystems. Changing in shifts in their phenology can have cascading effects to other organisms and other systems, such as mismatches between plants and the animals that rely upon them in mutualistic relationships, or animals that eat them, or between plants and changes in ecosystem carbon source sink dynamics. However, many gaps remain in our knowledge of plant phenology. There's a general kind of paucity of long-term observational data sets, and the information that we do have comprises a bunch of biases. As you can see on the top panel here, most of our phenological data and studies of phenological events across the landscape come from temperate regions of the world. And our recent reliance on remote sensing technologies have left various spatiotemporal gaps in our knowledge. We have data at certain spatial and temporal grains. We have data of certain systems, but not across the broad biodiversity that exists on our planet. These kinds of studies are often focused on a small subset of species or functional types within particular assemblages, such as a dominant woody species, because they're conveniently observed. Finally, despite many informative studies, we still know comparatively little about intraspecific variation in phenological responses. Along these lines, and I don't think I need to convince anyone in this audience, herbarium specimens represent a broad sampling of evolutionary diversity across large, extensive space and time. So here on the top row, you can see photos that I took from the field, and each of these photos represents a distinct phenological phase, such as the fronds unfurling here on this fern. This trillium is in flower. These leaves have changed color and senesced, and this mustard is fruiting. On the lower row, you can see images of herbarium specimens that have captured these same events, demonstrating their usefulness for phenological studies. In particular, of particular interest to me was the fact that these specimens comprise information about reproductive events such as flowering or fruiting. So many plant specimens are collected with reproductive organs on them, together with information on when and where these events occurred. This allows us to infer the environmental conditions under which certain phenological events happen. With increasing digitization, these resources have become increasingly easier to access in large volumes. Millions and millions of plant and animal specimens have been digitized in various ways, and these images and other types of media are largely available online. This has enabled digital-only workflows to be used to address a wide range of questions, including those of phenology, which I'm going to be talking about today, but other fields as well. However, extracting useful information, such as the presence of flowers or fruits, from millions and millions of very different digitized specimens can still be a major challenge. To facilitate such efforts, we developed CrowdCurio, an online crowdsourcing platform for image annotation, in a collaboration with scientists at the University of Waterloo and Harvard University. Now, citizen scientists are presented on this platform through Amazon Mechanical Turk with digital images of herbarium specimens and examples here of the reproductive organs they are asked to count. They count these organs by placing, scrolling around, zooming in and out of these specimen images, and placing little round markers where they observe, say, buds, flowers, or fruit. Now, this allows us to quantify the number of different reproductive structures and infer the phenological stage the plant on the specimen was in when it was collected. The locations of these organs are also stored for future analyses and some other projects that I might not be talking about today. So these graphs quickly look at the correlation between citizen scientists' counts of reproductive organs and those of expert botanists for flower buds, flowers, and fruits. And here you can see that the counts correlate very strongly. The two different colors here are for two different species, and the R values in both cases were all above 0.9. Therefore, we're able to reasonably verify that crowdsourcing is a useful method, an efficient method for phenological data collection from specimens, even when the people collecting these data are not necessarily trained botanists. Harnessing the power of crowdsourcing in this way allows us to efficiently tap into kind of the vast wealth of digitized herbarium specimens that have been collected across the world throughout hundreds of years and conduct phenological studies at unprecedented spatial, temporal, and taxonomic scales. Okay, so using this tool, we assessed variation in plant phenological responses to climate across 30 common flowering species in the eastern United States. Now, these species had broad ranges that spanned a diversity of environments across a latitudinal gradient. And here you can see they span a good diversity of temperature as well as elevation. And their ranges can be divided into a couple of domains or climate zones, depending on how you want to delimit these environments. Now, today I'll be talking a bit about phenological sensitivity, and it's illustrated here in the lower left panel. Phenological sensitivity to climate, in this sense, refers to how much the timing event of an event, such as flowering or fruiting, shifts in response to changes in climate, in this case, spring temperature. Now, we examined thousands of specimens of these 30 species that span diverse life histories and found that most species generally respond negatively to spring temperature. That is, their phenological sensitivity to temperature is negative. You can see most of the values here depicted in different colors all fall under 0, meaning that their phenological sensitivity to temperature is negative. And this means that plants tend to flower and fruit earlier with warmer springs. However, we found, we also found a great degree of variation in how sensitive these plants' phenology was to temperature. And you can see that these dots have very differing values among species. And an interesting trend here is that we see plants on the left end. These are things that tend to flower and fruit earlier. These species tended to be more sensitive to spring temperature than species that flower later and fruit later in the year. Now, our approach allowed us to compare within species and between region sources of variability for both mean flowering time and its sensitivity to climate. And surprisingly, we found that as much variation in phenological sensitivity exists within species as it does between different species. On this graph, you can see that the variance we find between species in flowering and fruiting is on par with the variance we find within species. This means that different populations of the same species can drastically differ in their phenological responses to temperature. So how does this variation manifest across the landscape? We found that phenological sensitivity to temperature is generally greater at lower latitudes in warmer climates. So if the temperature were to rise uniformly across a species range, chances are southern populations would advance their flowering and fruiting to greater degrees than northern populations would on average. We believe that plants in these southern latitudes tend to be more fine-tuned to temperature cues because warmer, predictable climates reduce the risk of reproducing in non-ideal conditions. For instance, in the Northeast, weather tends to be a bit temperamental, and it's often snowing after stretches of warm days in early spring. In New England, people say that if you don't like the weather, just wait 5 minutes. And this kind of means that temperature cues may not be the most reliable thing to fine-tune your phenological timing upon. So urbanization is another factor that could introduce variation in phenology across species ranges. The world is increasingly becoming urbanized, and no surprise, most of the world's population lives in these kinds of urbanized areas. And the result has been that increasing numbers of plants and animals are being exposed to anthropogenic disturbance, with their habitats becoming greatly modified by human activity. And urbanization has been shown to affect plant phenology through the what we call the urban heat island effect, where urban areas generate heat, making them a bit warmer than the surrounding more rural areas. This warming effect has been implicated by previous studies in advancing the growing season of plants that are in urban areas or near urban areas compared to those that are in less urban or rural regions. Now, using the same approaches we outlined above, we gathered phenological data for 200 diverse species across a gradient of climate and population density. Population density here is used as a proxy for the degree of urbanization an area has experienced, and it's been used as a proxy in many other previous studies. And because herbarium specimens have been collected across wide spatial and temporal breadths, they not only represent a wide sampling of climate and kind of environmental factors, they also represent, a diverse sampling of different population densities or degrees of urbanizations through space and time. Okay, so first we found that a higher population density was associated with accelerated flowering, especially in colder areas, as evidenced by this effect plot and the little snowflake here. These lines represent the phenological responses to population density after accounting for climatic effects. And warmer or redder colors indicate warmer areas. So this result is consistent with prior studies that have examined the influence of urban heat island effects on plant spring phenology. So this heat island effect could facilitate reaching the necessary degrees of thermal accumulation for initiating plant growth and reproduction in cold winters, right? And that's why we see that it advances plant phenology in cold areas. However, in warmer places, it could also reduce the chilling accumulation required for plants to break dormancy in warm winters and increase drought stress. And these factors can act to delay flowering and fruiting in certain conditions, as we find in our modeled estimates here. This trend is actually evident in the underlying herbarium data when we examine it directly. For instance, here are two common species across the eastern United States. We have Jack in the pulpit, Aracema trifilum, and Sisyncreum, the eastern blue-eyed grass. And in both cases, you can see that these guys flower earlier in response to increasing degrees of urbanization more so in the northern, colder part of their ranges than they do in the warmer parts, the warmer, more southern parts of their range. Now, previous studies of the effect of urbanization on plant phenology primarily focused on temperature. However, our results suggest that precipitation also has a big role. Precipitation can also mediate the effects of urbanization on plant reproductive phenology, in that we found that higher population density was associated with accelerated flowering in wetter areas, as depicted by this dark blue line, and the drop of water as compared to drier areas, as depicted by this cactus icon. So urbanization is often associated with the heat island effect, as we mentioned earlier, but it's also associated with increased drought stress due to many factors, including the increase in impervious surfaces. And it's been shown that drought can delay the development of plants and their reproduction. So we might hypothesize that in areas or years that receive less spring precipitation, plants might experience elevated levels of drought stress with increased urbanization, which can delay the development of flowers and fruits. On the other hand, increased precipitation could offset these effects. They could offset urbanization and increased drought stress and associated phenological delays, allowing plants to accelerate their development in response to the warmer ambient temperatures that are also associated with increased urbanization, as we saw in the previous graph. So what does this mean for the plants? One thing we can look at is frost risk. So urbanization can also shift the date of last frost. This is the last day at which frost is observed. And as you can see in the lower panels, unlike flowering time, the last day of frost always advances with increasing population density. The slope of all of these lines is negative, regardless of the temperature or precipitation conditions of the area. Thus, in general, we found that urbanization advances the end of frost faster than flowering in most conditions. With global warming, the frost dates are predicted to advance at a generally faster rate than the flowering of the plants that we looked at. However, this did not lead to reduced frost risk across the entirety of these species ranges. In fact, our results demonstrated that urbanization could also increase frost risk under certain circumstances. Because flowering phenology tends to respond differently to increases in human population density across their ranges, they can at times advance faster than the last frost date, especially in colder areas and cold and wet years, as you can see in these choropleth maps, where red indicates increased frost risk under climate change. The numbers here represent the number of counties and the number of species, respectively, in each category. So in summary here, we showed that the effects of urbanization on plant reproductive phenology and frost risk can significantly depend on both temperature and precipitation, and that the phenological consequences of increased urbanization will be, well, pretty heterogeneous across the landscape, but likely not random. So we just went over how variation in phenological responses to urbanization can lead to increased frost risk in certain areas and decreased frost risk in others. So this is kind of another piece of evidence that shows that there is immense variation in species phenological sensitivity to climate across their ranges. This graph is taken from a slide before, where we previously noted that there is as much, again, variation in the phenological sensitivity to climate between species as there is within species, right? So let's talk a little bit more about the consequences of such variation. The left column here depicts flowering dates for numerous congeneric species across the eastern United States. The second and third columns depict their sensitivity to spring temperature and precipitation. And the last column depicts the residual time trend of flowering phenology, how it's changed over time. And I know this graph is pretty busy and a bit hard to read, but what I want you to just notice here is the fact that you can see that flowering time and its response to temperature is very diverse, right? It's all over the place. There's a lot of variation across species, even very closely related species in the same genus. And you can see that because there's a lot of variation in the phenological timing and its sensitivity to climate, the way these plants have shifted their phenology through time is also very diverse. So here we can surmise that because flowering time and its sensitivity to climate varies greatly within and across species, climate change can alter both intra and inter specific relationships. Now, one way intra and interspecific variation in phenological sensitivity could manifest is what we call phenological character displacement. So character displacement refers to when, at least in the context of phenology, it refers to when species exhibit different phenologies when in sympatry versus when they exist apart. For instance, you can see here on the left panel, species could flower further apart in time when they are in sympatry versus when they do not occur together to partition resources or reduce competition. Alternatively, species flowering times could converge in sympatry due to facilitative interactions, density-dependent effects, or environmental or evolutionary constraints, which is illustrated here in the right panel. Now, we set out to assess the presence of phenological displacement among sympatric congeneric angiosperm species by comparing the results of two phenological models. The co-occurrence model, one that takes into account sympatric co-occurrence among closely related species, and the null model does not use this information. And then we compared the estimates of these models to see if there was any difference, if taking into account co-occurrence among closely related species changed our expectations of when these species would flower. Now, despite examining over 100 species in 20 families, pretty much spanning the entire diversity of the angiosperm tree of life, we didn't find much evidence of phenological displacement. You can see that here in this density plot, the estimations of both models largely overlap. These different colored dots and bars are also showing the same results averaged across the different genera of species that we examined. We did find, however, that flowering seasons in the eastern United States are likely to lengthen in the future, and that with climate change, overall, the flowering times of congeneric species will drift apart. Here, in the left two panels, you can see the kind of average season length of 1985 and how this length will change by 2055. And you can see again, all the values are positive, indicating the flowering season will expand for all of these species across their ranges in the eastern United States. On panel C and D, you can see that the flowering gap between congeners, regardless of whether they are only a few days to months, is almost uniformly going to increase with climate change. So species, congeneric species, many of which can actually hybridize and exchange genes, are going to diverge in their flowering times in the future. Now, this will likely alter patterns of gene flow and have interactions with mutualists and competitive interactions among taxa, but more focused work is needed to assess the kind of specific ecological and evolutionary consequences of flowering time divergence among closely related co-occurring taxa. Okay, so along those lines, let's take a more focused, relatable look at the potential ecological consequences and and interspecific variation in phenological responses to climate. The photo here is one I took in a forest nearby where I live, and show a bunch of Virginia spring beauties. Now, these guys are super common around here in Indiana and in Illinois. And I think we're about at the time of year when we should be seeing these guys popping up in the understory of forest in massive numbers. My postdoc, Dr. Yingying Shi and Hannah Thamabong, an undergraduate in the lab, conducted a case study on the phenology of these spring beauties and the minor bees that pollinate them. Hannah's also something of an artist, so you'll see little drawings of the flowers and bees in the next several slides that she drew. Okay, so let's talk a little bit more about this case study, the system we're using here. The Virginia spring beauty, or Claytonia virginica, is an early spring ephemeral that comprises floral color morphs ranging from kind of white colors here, as you can see in the two images here, to more pink flowers, as you can see here. Now, these two color morphs often occur in sympatry in the same populations, but it's not certain how these color morphs are maintained. There's some evidence that there might be slight differences in pollinator preference, and there could be divergent selection by pathogens versus herbivores on different flower colors. And these kind of selection pressures might play a role in maintaining these flower colors, but not much has been done or examined in regards to how their phenologies might differ. Now, these flowers are pollinated by one Andrena originae, the spring beauty minor bee, which is a specialist pollinator, and it's the solitary ground nesting oliglectic bee that relies solely on Claytonia virginica to successfully reproduce. It's been noted that these guys mate on the leaves of or flowers of Claytonia virginica, and they also need the pollen from these flowers to make little pollen balls to lay their eggs in, which the larvae eventually eat after they hatch. In this study, we used data from specimens as before, though this time we included insect collections, insect specimens here, like this, specimen of Andrena originae. That was, I forget where this is from, but that was collected in a museum and we used in our study. In addition to specimen data, this time we also utilized data from iNaturalist, which many of you might be familiar with. iNaturalist is a citizen science platform for mapping and kind of sharing observations of biodiversity across the globe. An added advantage of iNaturalist data is the fact that they're almost always paired with photos of the organisms here in real life. And this allows us in this case to determine the color morphs of the flowers that were observed. Also, because iNaturalist started out, I think, in 2008 and became popular with the spread of smartphones, most of these photos have fairly accurate coordinate data, unlike many of our historical collections, allowing us to kind of exactly pinpoint where these species and color morphs are occurring. A downside to these data are the fact that unlike the majority of natural history collections, iNaturalist data are collected opportunistically by people with varying levels of botanical knowledge. And therefore, these guys can be more prone to misidentifications and errors. In fact, I've seen an observation of what the observer touted to be yellow star thistle, but it was pink and obviously wasn't yellow. So there can be many errors in this data, so you have to be careful in utilizing them. So before we actually analyzed any of these data, we wanted to make sure that iNaturalist data was not too bad. And one way we did this was to compare the ranges of Virginia spring beauty and its pollinator as inferred by iNaturalist data, and compare it with their ranges inferred by specimens. And we found that those ranges were largely congruent. And we also examined all the images and weeded out any obviously erroneous records. And finally, this system is pretty amenable to this approach because Claytonia virginica is very distinct. It's a pretty well-known and easily identifiable member of the woodland flora. And its pollinator, Andrena eriginae, because it's a specialist, is almost always observed and photographed on the plant itself. So it's a system where we would likely see less errors than some more difficult plants or insects. So you can see in these graphs, again, that the ranges of Claytonia virginica and Andrena eriginae all are largely congruent regardless of the data source used to infer them. And you can also see that the ranges of the white and pink color morphs overlap almost perfectly, indicating that these guys indeed do often occur in sympatry. So based on these data, we first just simply assessed the phenology of color morphs and their pollinator using kind of 5-kilometer grids where they all co-occurred. So based on these 5-kilometer grids, we found that where both color morphs co-occur, the white color morphs tend to flower slightly earlier. So this dotted line represents a perfect correlation, and the actual line is slightly above it, where pink flowering date is the y-axis and white flowering date is the x-axis. So anything above the dotted line would indicate that the pink color morphs are flowering a little bit later in our observations. We also found that bees tended to be observed a little later than both of these color morphs. Again, the lines here, both of the regression lines are below the dotted lines, which represents a perfect correlation. The bees seem to emerge or are active in flight about 4 to 9 days on average after we observe flowering. And this actually, given the biology and the phenological progression of Claytonia, this might actually correspond with peak flowering across spring beauty populations. Interestingly, the phenological sensitivities, the phenological responses of these organisms to climate showed opposing relationships between spring beauties and their minor bees. So both color morphs of Claytonia were more phenologically sensitive to temperature in warmer areas, hence, which is illustrated by the fact these lines have negative slopes. So where winter temperature is warmer, these guys tend to be more sensitive to climate. They tend to respond more strongly in their flowering time to changes in climate. White color morphs were slightly more sensitive to temperature on average than the pink color morphs were. However, the really interesting thing here is the fact that the minor bees' phenological sensitivity shows an opposing relationship to temperature. Here, the minor bee is more sensitive to temperature in warmer areas and less sensitive to temperature in colder areas, which is the exact opposite trend we're seeing with Claytonia virginica. Now, due to these opposing trends, Claytonia virginica phenology tended to be more sensitive to temperature than the bee in warmer areas down here, while bee phenology was more sensitive to temperature in colder areas than the flowers it pollinates. So again, what we found is that flowering tends to advance with warming, congruent with many other studies of plant phenology. And flowering time tends to respond more strongly to changes in temperature in warmer regions, as we found in our larger scale results. Now, white color morphs exhibit a slightly greater variation in phenological sensitivity to temperature than pink morphs. And spring beauty minor bees also tend to advance their activity with warming. However, in contrast with the flowers that they pollinate, bee phenology responds more strongly to temperature changes in colder regions. With climate change, this phenological variation is predicted to result in heterogeneous shifts in the flowering time of the two morphs of Claytonia virginica. So you can see here on this map that the predicted changes for flowering phenology of the two color morphs are not really spatially congruent. The areas where one color morph is expected to experience large advances does not always correspond to larger advances in the other color morph. And these heterogeneous responses to climate change result in really varying degrees of expected change in the temporal gaps among these two color morphs. And you can see here, these numbers are for each ecoregion. And you can see here that in some ecoregions like this one, the temporal gap between white and pink flowering is expected to increase in all the 4 different climate change scenarios that we examined. In other ecoregions like 9.2.3 down here, they're expected the temporal gap between the flowering of white and pink color morphs is actually predicted to become reduced. They're going to flower closer together in the future. Now, these changes, again, are likely to alter gene flow among these color morphs, but the consequences on the maintenance of color morphs and their fitness are kind of difficult to predict with the data we have here. Likewise, the spring beauty minor bee's phenology will shift in various ways across its range. You can see here that larger advances are predicted to occur in the colder parts of its range with climate change, where, if you remember, the spring beauty minor bee is more phenologically sensitive to climate. Now, this will result, again, in various changes in the temporal gap between the minor bee and the flowers that it pollinates. But unlike the gap between color morphs, here we see a single direction, almost a single direction across the board, in that, in general, climate change will result in increased temporal divergence among Claytonia virginica and its pollinator across most of the regions we examined. Overall, most of the southern ecoregions were under higher risk of phenological mismatch with warmer winters, where Claytonia tended to show greater phenological sensitivity to temperature, much greater phenological sensitivity to temperature than Andrena. Also, we saw that the flowering phenology of white color morphs was predicted to be to diverge further from bee phenology than that of pink morphs. As you can see here, that on average, in most cases, the blue bars are taller than the pink bars, which respectively represent white and pink color morphs. So in summary, we found that temporal divergence among color morphs of Claytonia virginica and its pollinator is likely to happen in most regions with climate change. And white color morphs and southern regions are under higher risk of plant pollinator mismatch. We also found that the negative impacts of phenological mismatch may be more immediate and severe for the short-lived univoltine oligolectic bee species, because Andrena eriginae, as I just said, is short-lived. It only reproduces once every year. So it only has one chance or one flowering season in which to successfully reproduce. Whereas spring beauties are perennial and can reproduce multiple times throughout their lifespan. And further, they, especially in the southern parts of their range, they may be pollinated occasionally by other species such as bee flies. Even though these minor bees tend to be their primary pollinator and their near sole pollinator, especially in the northern half of their ranges. So this kind of demonstrates that variation in phenological responses across multiple axes need to be incorporated to fully understand how ecological interactions will change in the future. And we need to treat species as not kind of uniform entities in terms of their phenological responses, but things that have varying phenological responses across their ranges. So I hope I've convinced you that there is large intra and interspecific variation in plant phenology and its sensitivity to climate, and that this variation can manifest in diverse ways. We saw that urbanization effects on plant phenology are not uniform across species ranges, but are mediated by both temperature and precipitation. We also observed that climate change can cause the flowering times of closely related congeneric species to diverge in the future. Finally, we demonstrated that climate change can result in very heterogeneous patterns of phenological mismatch within and among interacting species. With that, I'd like to acknowledge my collaborators. All the work I presented today was part of a team effort and would not have been possible without these people, as well as the many museum collectors, curators, and citizen scientists across the world. Thank you all for listening and coming to my talk, and I'd be happy to answer any questions that you might have.
Thanks very much, Daniel. That was great. We've got a few questions here. First one is from Akila Wilkes, and you actually answered one of her questions. She was wondering if your work covered the impact on pollinators, and then and then you got a little bit later and answered that. But the other question Akila had was, do you know if other disciplines have easy access to citizen science like you?
Yes. So I think the whole citizen science thing didn't start with phenological studies. It didn't even start with plants, as with most things. I think citizen science approaches were pioneered with animals. So there's a platform called Zooniverse that's been very popular. And on there, you can see various crowdsourcing projects, including like looking at identifying animals that come to water holes in Africa, or things like that. There are also many other crowdsourcing kind of studies that include, or citizen science studies that are not even in the realm of like the biological sciences, but are things like more computer science oriented, where they're trying to generate training data for machine learning algorithms. For instance, the CAPTCHAs that sometimes you do are likely feeding into some of these projects where you have to type in a line of text from a picture of a passage in a book or something, or when you identify traffic lights or animals or boats or cars. So I guess, yes, many other disciplines do have access to citizen science, and many people have, like us, have been taking advantage of the Amazon Mechanical Turk platform, which can connect a person and his or her tasks with the people who want to do those tasks, obviously for money, usually. but it makes the whole process a bit more approachable and feasible. So you see hundreds of tasks, thousands of tasks on Amazon Mechanical Turk, anything from random surveys to more kind of scientific and involved endeavors, if that answers your question.
Great. Yeah, I think that answers Kila's question. Yeah, that's awesome. Thank you. So then we have a couple questions from Rick Ree here. First one is, what's the genetic basis and biological or ecological significance of pink versus white in Claytonia?
Great question. So based on several lines of evidence, it's been suggested that the floral color morphs do indeed have a genetic basis and are controlled by a few genes, but no one's directly looked at if that is true or what genes are involved. The chemicals involved are a mouthful, and I can't properly say them or remember them right now, but the white versus the pink coloring is determined by a couple of chemical compounds that are also have been hypothesized to be involved in resistance to herbivores and pathogens. And if you look at a paper by Frey in, I think, 2004 and or 2007, they hypothesized that these chemicals are inferring different degrees of resistance to herbivores and pathogens, and they correlate with the color of the flowers. And that might be one of the reasons why we keep seeing both flowers maintained instead of having something in the middle take over everything. So, and on an ecological perspective, the color of the flower seems to be maintained regardless of soil type. And people have grown these guys in the greenhouse, and they also seem to maintain the flower color in these conditions. So they're not changing based on the environment, at least.
Okay, great. Thanks. And then we've got a follow-up here from Rick, or a separate question. What assumptions about collecting effort are needed to ascertain the effect of variables like temperature on phenology?
So I'm not quite sure I understand the question, but in general, we always assume that our collections are biased. We know that they're biased. We've done research on how biased they are, and so one way we do try to account for these biases is to first remove erroneous records, but also remove duplicates and kind of coarsen our data. So in these analyses, not all, but most of the analyses I talked about today, we've coarsened the data to basically the county level across the eastern United States. This isn't ideal, but it does help deal with some of the biases in sampling effort that have occurred across the landscape. This is probably less feasible when you work in the West Coast, where some counties are as big as states, small states. And the kind of fuzziness and uncertainty around these collections can be accounted for in terms of temperature or precipitation or any climate variables in a couple of ways. One way people commonly do it is just to take the kind of centroid value. That's not what we do, because they're often not representative of the geopolitical entities in which these collections are made. But we've taken means or averages or median values of climate at coarser scales. We've also incorporated at times the degree of environmental heterogeneity that is associated with each cell or each county, for instance, so that we can weigh the uncertainty in the climatic factors that have been extracted from these general areas where the specimens have been collected. I hope that answers your question, but I'm happy to go into more detail if you want.
Great, thank you. And I was just wondering, did I understand correctly that the spring beauty and its pollinator are both specialists on one another?
I would say near specialist. So in the northern part of its range, it seems like Claytonia virginica is almost solely pollinated by Andrena originae. In the southern part of its range, people have observed that bee flies sometimes seem to be pollinating them. And occasionally, there are other insects that have been observed on the flower, but people who have looked at the insects noted that there's like not a lot of pollen sticking to them, suggesting that they're not being efficient at pollinating these guys. The spring beauty minor bee, on the other hand, does seem to be almost like 100% reliant upon Claytonia virginica, regardless of the northern or southern part of its range. It's all the pollen balls that people have found in their dens are from Claytonia virginica. Again, they seem to mate on the plant. Whenever you find photos of them or you capture them, they were on the plant.
So are there any thoughts on what will happen if this the kind of phenological discordance increases? Is it kind of curtains for the minor, or do you think it'll switch to an alternate source plant?
I think it could be possible. I mean, there's a closely related species. There's Claytonia caroliana, which co-occurs or occurs near the species sometimes, but I don't know. It seems pretty bad for the bee. Its life cycle is pretty tied into this one species of plant, and yeah, it doesn't look good for it.
Got it. All right. Well, I think that is all we've got. So thanks again very much, Daniel. That was wonderful. Thanks everyone for coming out. Yeah, we'll see you next week. Thanks again, everyone. Take care. Bye.